This is the MV Hondius, a luxury expedition ship. In April 2026, it departed Ushuaia, Argentina, carrying 147 passengers and crew on an Antarctic voyage. Navigating these harsh, remote environments means operating hours, if not days, away from immediate medical infrastructure. The vessel is entirely isolated. Midway through the journey, multiple passengers began crashing. Initial symptoms of fever and gastrointestinal distress progressed rapidly to pneumonia, acute respiratory distress, and eventually hemodynamic shock. The consequences were severe. 3 people died. Others required critical ICU medical evacuations, and as the situation spiraled, the ship was refused entry to port in Cape Verde. Medical teams quickly ruled out influenza and COVID-19. When the laboratories finally sequenced the pathogen, the results confirmed a highly lethal threat, the Andes Orthohantavirus. Welcome back. I am Dr. Urvish Joshi. If you are a medical undergraduate or a postgraduate in community medicine preparing for your exams, this is a real-world case study you must understand. Today, we are breaking down the specific virology of this pathogen, India's IDSP reporting framework, and the international health regulations governing the global response. This outbreak represents a unique stress test. It is the collision of a rare, highly lethal pathogen with the speed and closed system nature of modern international luxury travel. This electron micrograph shows the spherical structure of hantavirus particles. They are enveloped, tri-segmented, negative-sense RNA viruses belonging to the family Hantaviridae. This map illustrates the geographic and clinical split. Old World strains in Europe and Asia cause hemorrhagic fever with renal syndrome. New World strains in the Americas cause hantavirus pulmonary syndrome, carrying an extreme case fatality rate of 35 to 50%. The standard transmission vector for these viruses is classically zoonotic. Human infection occurs strictly through the inhalation of aerosolized rodent urine, feces, or saliva. The Andes strain is endemic to the Patagonian regions of South America, near Mount Fitzroy seen here. However, Andes possesses a specific biological anomaly absent in other hantaviruses. It can achieve sustained person-to-person transmission. By bypassing the need for environmental rodent exposure, the virus turns an infected human into a mobile vector for a pathogen that kills up to half of the people it infects. Proof of this transmission mechanism comes from the ARG epuyen isolate, sequenced from human samples during a 2018 Argentina outbreak. Genomic analysis reveals mutations in the nucleocapsid and small non-structural proteins, driving human transmissibility. On the exterior, we target the envelope's GN-GC tetramers. Here, the GC glycoprotein drives the virus from membrane hemifusion to complete pore formation in host cells. These molecular adaptations enable the virus to achieve the high viral loads necessary for transmission during prolonged, close physical contact, allowing the pathogen to move between humans and shared spaces like a cruise ship cabin. Epidemiologists face a compounding variable. The Andes virus has a highly variable incubation period of 1 to 8 weeks. Because microscopic viral mechanics allow for long-term silent incubation combined with human-to-human spread, public health authorities cannot rely on standard outbreak timelines to track and isolate the infected. When an outbreak breaches a closed system like a cruise ship, containment depends entirely on the domestic systems ready to catch it, especially if an infected passenger lands in a heavily populated nation like India. This represents India's Integrated Disease Surveillance Program. The IDSP operates as a decentralized 36-state safety net designed to detect and respond to epidemic-prone diseases. Looking at the official protocol, memorize these mechanisms. The framework uses an S form for suspected cases and a P form for presumptive clinical cases. The L form demands laboratory confirmation, linking symptoms to sequenced genetic data. Managing the Hondius outbreak requires international coordination, led by the World Health Organization at their headquarters seen here. This global response is mandated by the International Health Regulations of 2005. It places a legally binding obligation on all member states to develop and maintain the core capacities to detect, assess, report, and respond to threats. The IHR emphasizes points of entry. Airports with thermal screening serve as primary choke points to intercept international spread. Biological lethality is only half the threat. The real danger lies in the catastrophic breakdown of these frameworks. If IHR and IDSP fail to communicate, a clinical diagnosis remains an isolated incident instead of a national alert. Let's review the high-yield takeaways. First, Andes virus is the only hantavirus with person-to-person transmission and a 1 to 8 week incubation. Second, severe pathology, like shock and pulmonary edema, is driven by GC glycoprotein mutations. Third, containment requires strict IHR compliance and rapid IDSP reporting. As someone deeply interested in AI and health research, I want to share the methodology behind today's breakdown. To synthesize this case study, I utilized AI tools to rapidly process over 100 global epidemiological reports, including specific threat assessments like this brief from the ECDC. In an environment defined by high data velocity, artificial intelligence serves as a mandatory productivity booster for doctors tracking global risks. If you found this breakdown useful for your community medicine prep, hit subscribe. Leave a comment below telling me which emerging pathogen or health framework we should decode next. Effective global health depends on both the microscopic science and the macroscopic policy. Mastering both allows medical professionals to identify threats and mobilize the systems designed to stop them.